Diabetes is associated with cardiovascular disease and accelerated atherosclerosis. Vascular smooth muscle cell activation is a key process that drives inflammation and contributes to the development of atherosclerotic plaques. Normally, inflammation is resolved by the production of endogenous modulators, including lipoxins like LXA4. However, failure to resolve these inflammatory processes might contribute to the pathogenesis of atherosclerosis and diabetes. Platelet-derived growth factor, or PDGF, upregulates the expression of several genes involved in vascular smooth muscle cell activation. The researchers used quantitative reverse transcription PCR, or qPCR, to assess if LXA4 could attenuate PDGF-induced upregulation of 9 of these genes in vascular smooth muscle cells.
The experiment included 4 treatment arms. Mouse VSMCs were treated with a vehicle-controlled PDGF only, LXA4 only, or PDGF and LXA4. Reverse transcriptase qPCR was then performed on total RNA extracted from VSMCs in each group. Details of the reagents, primers, and probes were provided in the supplemental information. However, the methods failed to specify whether fluorescent signals were normalized to 18S RNA or GADP-H for the result illustrated in Figure 4a.
The data showed that LXA4 reduced PDGF-induced upregulation of several of the genes of interest. A strong figure legend includes a brief description of the experiment. However, in Figure 4a, the legend title was vague as it did not give an overview of the experiment, the technique used, which was RT-qPCR, statistical analysis used, the vehicle control and its concentration, or redefine the abbreviations. In addition to that, it did not specify what internal reference gene the y-axis of the figure is normalized to. Additionally, lines are not used to show the significance between groups. The authors indicate that LXA4 might prevent atherosclerosis as it inhibits PDGF-mediated vascular smooth muscle cell activation. This claim is justified because based on qPCR, LXA4 reduced the expression of several genes that PDGF upregulates to induce vascular smooth muscle cell activation.
Overall, qPCR was effectively used in this study. However, the reproducibility is threatened because it was unclear which housekeeping gene was specifically used for normalization. Furthermore, the figure legend is lacking important information, which forces readers to continuously refer to methods to interpret the figure. Finally, interpretation of Figure 4a was made more difficult by the author's failure to use lines to demonstrate the significance between groups.